Today, we're diving into a classic whodunit, a real microbial detective story. You've got the victim, the human nervous system. You've got the crime scene, a simple wound. And you've got your prime suspect, a bacterium. But, like any good mystery, the real culprit is not who you think it is.
So, if I asked you to name the villain behind tetanus, you'd probably say the bacterium Clostridium tetani. And, well, you'd be partly right. See, it's at the scene of the crime, for sure, but it's not the one actually pulling the trigger. The real killer is something much, much more insidious. So meet the true villain of our story, a neurotoxin called tetanospasmin. The bacterium itself, it's surprisingly non-invasive. It just hangs out in the wound, kind of like a quiet little factory. But that factory is pumping out this incredibly dangerous weapon, the toxin. And that's what travels through the body, causing all the devastating damage we know as tetanus. All right, so to solve this case, our first step is to get a really clear description of the suspect, the one that's producing this toxin. What exactly are we looking for when we peek under the microscope?
Here's the rap sheet. It's a gram-positive rod-shaped bacterium, and it's an obligate anaerobe, which is just a fancy way of saying it absolutely hates oxygen. But its most unique feature, our first big clue, is this terminal round spore at the very end. This gives the whole thing its famous drumstick or tennis racket kind of look, a very, very unique visual profile. So that leads to the obvious question, right? If our lab detectives spot this super distinctive drumstick shape in a sample from a wound, have they cracked the case? Is it time to close the file? Well, not so fast, because this is where our investigation hits its first major snag. It turns out our suspect has a doppelganger, an imposter named Clostridium tenomorphum. And the crazy part? It looks identical. It has the exact same drumstick appearance, but it's completely harmless. This means just looking at it, microscopy alone, is totally unreliable. Visual ID just isn't going to cut it here.
So since we can't trust our eyes, our detectives need to change tactics. It's time for a stakeout. We need to isolate our suspect and watch its behavior, see if it acts like a criminal.
Now, in the lab, the stakeout is a two-stage process. First, they put the sample into a special cooked meat broth. If our suspect is in there, it'll start to digest the meat, turning it black and producing this really foul odor. That's a huge behavioral clue. Then, they take that and grow it on a blood agar plate. And here, C. tetani reveals another telltale habit, a swarming growth that just spreads across the entire surface.
Okay. So now we've got a suspect that not only has the right look, but it has the right criminal behaviors in the lab. It's looking like an open and shut case. We're ready to make an arrest, right?
No, absolutely not. Because even with all of this evidence, the look, the smell, the swarming growth, we are still missing the single most important piece of the puzzle. We haven't found the weapon.
And that brings us to the final, definitive step in our investigation, the one test that can truly, without a doubt, confirm our diagnosis. We are now looking for the smoking gun. Let's just remember the core principle of this whole case. The disease is caused entirely by the toxin. So it really doesn't matter if we find the bacterium. What matters is proving that the specific bacterium we found is actually armed and dangerous, that it's producing tetanospasmin.
And we do this with something called a toxigenicity test. This is the ultimate confirmation. An in vivo test, often using mice, is done to see if the bacteria we've isolated can actually produce the toxin and cause the disease. This is it, the final, irrefutable piece of evidence that confirms a diagnosis of tetanus. Case closed. For real this time.
But our story doesn't quite end there, because in a brilliant twist, understanding our villain is actually the key to defeating it. The toxin itself has one more role to play in this story.
Science pulls off this incredible trick here. On the left, you've got tetanospasmin, virulent, dangerous. But by treating it with formaldehyde, scientists create the tetanus toxoid on the right. Now notice what changes. Its virulence drops to 0. It becomes totally harmless. But, and this is the crucial part, its antigenicity, its ability to be recognized by our immune system, stays high. It still looks like the enemy. And that is the absolute elegance of modern vaccines. We take the villain's primary weapon, we disarm it so it can't hurt us, and then we show it to our body's defenses. Our immune system then learns to recognize and fight the real weapon without ever having to face the actual danger.
So the very molecule that causes this devastating disease also holds the secret to preventing it. The solution was hiding inside the problem all along. It really makes you wonder, What other answers to our biggest challenges are right there in front of us, just waiting for us to look at them from a new perspective?